Hello, my name is Morgan Riley, and I'm a rising junior at The Ohio State University studying biomedical science. This summer, I had the privilege of participating in the NIH SenNet CUSP internship program at Dr. Ruhland's lab at the Oregon Health and Sciences University. My project was evaluating SASP proteins with single-cell resolution. Senescence is a state of permanent stable cell cycle arrest. Many different stressors can induce senescence, including DNA damage, oxidative stress, and telomere shortening. As you age, the number of senescent cells in your tissue increase, and these senescent cells release senescence-associated secretory phenotype factors, also known as SASP. These SASP include cytokines, ketokines, proteases, and growth factors, and are the main way in which senescent cells can communicate with their environment. To the right, you have a model of a senescent cell. As you can see, the SASP are released from the cell, and it also has increased lysosomal activity, which is associated with increased SA beta gal activity, which is one of the main markers of senescent cells used today. This is an image of senescence in the stroma. First, you have your healthy cells, and injuries inflicted upon these cells. This can be DNA damage, for example. This causes the cells to go into a state of senescence, and once they're in this state of senescence, they will release SASP. Some of these SASP recruit immune cells, and the immune cells are supposed to clear the senescent cells. However, as you age and you have more senescent cells and a weaker immune system, the immune cells cannot clear all of the senescent cells, allowing the senescent cells to remain and continue to secrete SASP. These SASP cause chronic inflammation in an immunosuppressed environment. This causes these senescent cells to be implicated in age-related diseases such as pulmonary fibrosis, osteoarthritis, and cancer. One of the goals of the SenNet Consortium is to create an atlas of senescent cells across the body in order to develop treatments that can target the pathological senescent cells. One of the challenges with this goal is that there's not one sole marker that can identify senescence. That is because senescent looks different between senescent cells across tissues. So how the phenotype of the senescent cells in your skin looks different than in your lungs. So my objective this summer was to identify proteins expressed by skin senescent cells in tissue with single-cell spatial proteomics. In order to complete this objective, we had a three-step workflow. Our model was a tamoxifen-inducible Cre recombinase FAST mouse. We had Cre positive and Cre negative FAST mice. And Cre is an enzyme that knocks out the LOX-flanked stop codon for P27 expression. When the stop codon is knocked out and P27 is overexpressed, it causes cell cycle inhibition and leads to senescence. In our control group, our Cre negative cells, we saw no senescence because that stop codon was not knocked out. Once we had our model, we treated the mice with tamoxifen for 5 consecutive days topically on their back. Then we harvested the dorsal skin and embedded it into CMC media onto pen slides. We then sent those pen slides off to the Pacific Northwest National Laboratory, where they completed laser ablation and droplet capture using NanoPET's technology. The NanoPET's technology allowed them to complete mass spectrometry at the single-cell level so that they were able to identify a list of differentially expressed proteins in senescent cells. Once we received that list of over 200 differentially expressed proteins, we chose 4 proteins of interest and completed the same process of treating the mice with tamoxifen, harvesting their dorsal skin, and embedding those into OCT media and onto glass slides. We then performed immunofluorescence staining for those 4 different proteins. We also validated these biomarkers in vitro, where we used skin fibroblasts from the FAST mice. We treated half the cells with tamoxifen and half were untreated to be kept as our controls. We performed SA beta gal staining and IF staining on these cells and captured the images. As I mentioned, we had over 200 proteins that were differentially expressed. We chose four, both a combination of known and novel factors. We chose S100A13 because it is a known inflammatory and pro-tumorigenic SASP factor. This allowed us to know that our staining was working correctly. We chose C3 because it's a novel factor in the complement system, PSMB6 because it's also novel and it's involved in protein degradation and the inflammasome, and Rab11 because it is involved in the exocytosis and endocytosis pathway. This is an important pathway that has not been researched in relation to senescence, so we were interested in looking into this protein. This is a visualization of our S100A13 tissue staining. We have our control and FAST group. As you can see, we have our green stain, which is our GFP stain. GFP is expressed at the same time as P27 is expressed, therefore it identifies our senescent cells. As you can see, there is an increase in green stain in the FAST versus control. The DAPI is our blue stain, and that identifies the nuclei, allowing us to visualize each individual cell. And the red stain is our protein of interest. In this case, it's our S100A13. The S100A13 is increased in the FAST as well, but what we were really looking for was the overlap of the green and red stain. In our last slide, you can see that composite image where we have an increase of the overlap in the FAST tissue versus almost none in the control tissue. So we were able to optimize this at a primary antibody concentration of 1 to 200 and secondary 1 to 800. For our PSMB6 staining, we saw the same decrease of GFP expression comparing from the FAST to the control. However, the red stain was uniform throughout. Based on the secondary alone picture, we were able to determine that the red stain was nonspecific staining by the secondary antibody. We tried this at different concentrations, but we're not able to optimize it further. Therefore, in order for validation, this will have to go further optimization. For a C3, we also were not able to visualize the difference in the red stain between the FAST and control. We tried this with 3 different primary antibody concentrations and 3 different secondary antibody concentrations. We concluded that S100A13 was validated in tissue immunofluorescence staining. However, while Rab11 was independently tested, neither Rab11, C3, or PSMB6 was able to be validated because it was ubiquitously detected across cell types, and it will need further optimization. At this point, we decided to move to validating the markers in vitro instead of in vivo. We were worried that the reason that the staining was not working was because of the tissue and it was blocking it. So when you're validating in vitro, you don't have the enzymes, the blocking media, and the vascular components inhibiting the antibodies. Therefore, the proteins of interest are more accessible, so we had greater faith that the staining would work properly. For validating these markers in fibroblasts, we first obtained the skin FAST mice fibroblasts. We then plated those cells and treated half of them with tamoxifen. We then waited 7 days and treated half of them with SA beta gal staining, and the other half immunofluorescent staining was performed with the same 4 antibodies. We found that there's significantly more SA beta gal positive cells in the tamoxifen treated cells versus the untreated cells. As you can see, the blue stain is the SA beta gal, and in the image to the right, there's a lot more blue cells than the untreated. We found there was 10% SA beta gal positive cells versus 30% SA beta gal positive in the tamoxifen. We found that there was 10% SA beta gal positive cells in the vehicle versus 30% SA beta gal positive in the tamoxifen. This allowed us to believe that our tamoxifen treatment did work, and we were able to continue on with our immunofluorescent staining. In the IF staining in vitro, we found that C3 is elevated in senescent cells. What we're looking for here is the red overlap with the blue. So as you can see, again, we have our blue, our DAPI stain identifies our cells. The red stain is our C3, and there's significantly more C3 positive cells in the tamoxifen treatment, with 92% of those cells detecting C3 versus only 35% of the vehicle cells were C3 positive. S100A13 was also elevated in senescent cells. We see an increase in the S100A13 expression in the tamoxifen treatment group, with 95% of those cells S100A13 positive, whereas only 42% of the vehicle were S100A13 positive. This validated our results that we saw in vivo as well. For PSMB6, we saw that 77% of the untreated cells and 85% of the treated cells were PSMB6 positive. As you can see, there's not much of a difference in the images from vehicle and tamoxifen, so this will need further validation. Rab11 was also detected in senescent and non-senescent cells. This is not surprising because Rab11 is involved in the exocytosis-endocytosis pathway. Therefore, a lot of cells express Rab11, and so we need to further optimize the stain in order to see the difference of protein expression in IF staining. Because here we saw 89% of untreated cells versus 100% of treated cells express Rab11. So these are pretty high numbers at the moment. In conclusion, we were able to validate S100 and C3 as biomarkers for senescent cells. However, the IF staining for PSMB6 and Rab11 needs further optimization. All of these stains will be repeated in order to be sure of our results. But in the steps for validating PSMB6 and Rab11, we're going to decrease the stress on the cells during the staining process to decrease the number of senescent cells in our control group. This could affect our results. We'll also try to adjust the antibody concentrations, and if that does not work, we will move to IHC and tissue staining. For our future directions, we hope to correlate experimentally induced senescence to natural senescence that occurs in aging. To start this, we performed SA beta-gal staining on aged mice versus young mice. These aged mice were over 2 years old, and we found that there was an increase of senescent cells in the aged mice versus the young mice tissue. What this means is that we will be able to generate proteomics data on the aged mice in the same process we did with the FAST mice. And from that data, we will be able to compare the list of differentially expressed proteins in the aged versus FAST mice. This gives us a more reliable and applicable list of differentially expressed proteins in senescent cells, because as I mentioned earlier, the type of stressor that induces senescence changes the phenotype of the cell. Therefore, if we have two different types of P27 and aging-induced senescence, and we can see the common link of those differentially expressed proteins, that will give us a better idea of biomarkers that are applicable for all senescent cells. I would like to thank the SenNet CUSP internship program and the NIH, as well as Elizabeth Martinez and KMT for allowing me the opportunity to participate in this research experience this summer and fostering my development throughout the process. I would like to thank all members of the Ruhland Lab and Pacific Northwest National Laboratory, especially Dr. Megan Ruhland, my PI, and Naomi Berkowitz, who I work directly under. All members of these labs helped me learn the intricacies of the research process, as well as develop skills that I would be able to take into my everyday life, as well as my future career.